औज़ुबिल्लाहिमिनश्शैतानिर्रजीम बिस्मिल्लाहिर्रहमानिर्रहीम रब्बि इश्रह ली सदरी व यस्सिर ली अमरी वहलुल उक़दतम मिन लिसानी यफ़क़हू क़ौली रब्बि ज़िदनी इल्मा वअल्हिक़नी बिस्सालिहीन अल्लाहुम्मा इन्नी अस्अलुका इल्मन नाफ़िअन व रिज़्क़न तय्यिबन व अमलन मुतक़ब्बलन. Assalamu alaikum students. Today you are going to learn about the enzymes inhibition. As you know, this is your chapter number 21, the biochemistry part number 5. Then afterwards you are going to learn about the types of the inhibitions, that what type of the inhibitors are there, and applications of enzymes. So we are going to start it with the enzymes inhibition. रोक देना, किसी भी enzymes की enzymatic activity को रोक देने का नाम. The process by which the activity of an enzyme is decreased or inhibited by the addition of certain substance is called the enzyme inhibition. And those who inhibit are called the enzyme inhibitors. An enzyme inhibitor is a molecule that binds to an enzyme and decreases its activity. By binding to enzyme's active site, inhibitors reduce the capability of the substrate and enzyme. There are three kinds of reversible inhibitors: competitive one, non-competitive or mixed one, and uncompetitive inhibitors. Competitive inhibitors, as the name suggests, compete with the substrates to bind to the enzymes at the same time. One by one you are going to learn about it. On the right side, if you observe over here, here is the competitive inhibitor. Next one is the symbols are used for the non-competitive inhibitors, and the third one, the symbols are used for the uncompetitive inhibitors. In irreversible inhibitions, an irreversible inhibitor will bind to an enzyme so that no other enzyme substrate complexes can form. Enzyme substrate का complex बन ही न सके. It will bind to the enzyme using a covalent bond at the active site, which therefore makes the enzymes denatured. An example of an irreversible inhibitor is diisopropyl fluorophosphate, which is present in nerve gas. It binds to the enzymes and stops nerve impulses being transmitted. An example of where we use the irreversible inhibitors in medicine is penicillin. Penicillin works by inhibiting the activity of the enzymes responsible for the creation of the bacterial cell wall. This means that water can enter the bacterial cell, causing it to swell, burst, and die, termed lysis. So splitting is there, irreversible inhibition. If you observe this picture, here is the enzyme, and then this one is the substrate. The lock and key model is going to be fitted like this, but whenever you will observe that irreversible inhibitors, they are there instead of the substrate. The inhibitor, the reversible inhibitors are there, and they stop the functionality. Then the reversible inhibition, a reversible inhibitor is one that once removed allows the enzyme it was inhibiting to begin working again. एक दफ़ा अगर remove कर दिया जाए तो enzyme अपनी activity दोबारा start कर लेता है. It has no permanent effects on the enzyme. It does not change the shape of the active site. For example, reversible inhibition may be competitive, non-competitive, or uncompetitive. Here, this is the enzyme, the substrate, the active site is there, then the competitive inhibitor, the substrate that competes for the active sites. Here, the non-competitive inhibitors which alter the formation of the enzyme, so active site is no longer fully functions. Next one is competitive inhibition. Competitive inhibition is interruption of the chemical pathway owing to one chemical substance inhibiting the effect of another by competing with it for binding or bonding. A molecule similar to the substrate but unable to be acted on by the enzyme competes with the substrate for the active site. Because of the presence of the inhibitor, fewer active sites are available to act on the substrate. For example, here is the enzyme, then this is the shape of the substrate, this is the shape of the inhibitor. Instead of the substrate, the inhibitor will take place the active site, and the inhibition binds to the active site, so no reaction directly blocks the active site, so no reaction will takes place. The process will stop. Now, in non-competitive inhibition occurs when the inhibitor can't bind to the active site due to the charge, shape, dissimilarities related to the substrate, but is still able to bind to the enzyme and cause a conformation, shape, charge change in said enzymes. So non-competitive means they are not competitive one, but here on the right side you can easily understand with the help of this picture that non-competitive inhibition. See, this is the enzyme, this is the active site, but here is the inhibitor. It attacks on the other side of the enzymes, but it will de-shape the active site, so the coming substrate cannot fit inside it. Inhibitor binds to a secondary site on the enzyme, and this change the shape of the active site and prevents the substrate from binding. So इस तरह इनका reaction जो है वो stop कर देते हैं. Uncompetitive one, uncompetitive inhibitions, also known as the anti-competitive inhibitions, takes place when an enzyme inhibitor binds only to the complex formed between the enzyme and the substrate. So, enzymes and substrate complex, the uncompetitive inhibition typically occurs in the reactions with two or more substrates or products. Overall, enzyme inhibition's mechanism is the competitive one here, the substrate inhibitor. Instead of substrate, the inhibitor will move and compete for the active sites. So इनमें जंग रहती है, दोनों में से कौन पहले पहुंचेगा. So इस तरह अगर inhibitor पहुंच जाता है तो substrate को जगह नहीं मिलती. So this is called the competitive one. They are competitive to each other. Non-competitive, they attack on the different side, but they will the substrate will not fix in it, and this is the non-competitive one. And in uncompetitive, it will de-shape the active site, so the substrate will not come and the reaction will not proceed. So in this way you learn about the inhibitors and inhibitions. Now the applications of the enzymes are there, where the enzymes are going to be in the see in the waste degradation, detection of the toxic pollutants, environment, promote the cleaner production, lead to the reduction. Industrial enzymes are the leather tanning, pulp and paper, starch and fuel, chemical and pharmaceutical, detergent ones, food and beverages, textiles, agro processing, animal feed additives, agrochemicals, and animal feed from the agricultural waste, etc. These are the enzymes which are used in the industries. In the biological industrial production, there are the biological detergents. Some enzymes are added to the detergents which are helpful in removing the protein and starch and stains them, and they are also able to remove fat, oil and the grease. इन सबको दूर करने के लिए biological detergents जो use किए जाते हैं. In fruit productions में for the extraction of the juice from the fruits require high temperature, but it affects the color and the flavor of the juice. So the cellulases and the hemicellulases which break up the cell wall, and all the liquid is released in the enzymes which are added to the fruits. In the production of ethanol, in the biofuel industry, the cellulose are used to break down the cellulose into the sugar, and which is then converted into the ethanol through the fermentation process. Then the high fructose syrup. Fructose is used as the sweetening agent. As you know, it is manufactured from the starch in the corn grains. And first the grains are converted into the starch slurry, and the enzymes amylase is added. Finally, the syrup is passed through a certain glucose isomerases enzymes, and then converts the glucose into the fructose. In this way, high fructose syrups are going to be useful. Then in the paper industries, as you know, in the paper industries, various enzymes like the amylase, xylinases, cellulases, and liginases are used for different purposes. Cellulases smoothen the fibers, amylase degrades starch to lower the viscosity and eating size and the coating for the paper. So in this way, these are beneficial for the paper industries. Overall, production of the microbial enzymes and the industry applications are. This is the overall view. Enzymes are the catalysts of the biological systems. They classify into oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. Sources of enzymes were animal, plant, and microbials. Amylase, techidases, first fungal enzymes used for the digestive disorders. Proteases, Detergent industries. Amylases and glucosidases is used in the glucose to form the starch. Glucose isomerases is the production for the fructose, and microbial renin for the cheese production. So in this way you learn about the applications of the enzymes in the as the industrial applications. So overall view that you learned today, the conclusion is that you learned about enzyme inhibitions and inhibitors, the types of the inhibitors, how they are going to be distinction from each other, what are their functionalities, and how they will affect the enzyme's activity. And afterwards we learn about the enzymes application where these biological catalysts are going to be used. In next lecture, inshallah, you are going to learn about the lipids, structure and classification of their classification and their properties, inshallah. May Allah be with you. Take care. Allah Hafiz.